All right. So I'm going to pull up my cheat sheet here. All right. So hello, everybody. Thanks so much for coming today. I'm so excited to introduce our speaker, Dr. Megan Phifer-Rixey. In her PhD work, she made important insights into how genetic heterogeneity is maintained in the wild and influenced by the environment using the flat periwinkle mollusk as a model system, which are adorable. She then began her postdoc working with my favorite species, with mice, especially Mus musculus, or the house mouse that many of us are familiar with from our lab. But many may not know that the lab mouse or house mouse species has multiple subspecies in the wild. House mice live in varied climates, from artificial New York City life to the completely wild prairies, from boiling hot arid environments to freezing cold northern climates. So how does a species that's so tiny and adapt to such incredibly different environments? Using a combination of field work, bench work, and modeling to reveal the secrets of wild house mice and speciation, Megan's work is giving us key insights into this important question. And using these subspecies also provides clues to unlock foundational evolutionary principles. I will let her tell you all about her exciting work that she's been doing lately, and I can't wait to hear what she has to say with us today. So without further ado, over to you.
Thank you, Emily, so much for inviting me to talk with you today, and thank you all for joining us this afternoon. I recognize that the novelty of Zoom seminars has worn off quite a while ago, so I appreciate that you're spending your time with me today to learn a little bit about my work in evolutionary genetics and house mice. All right. So before I even get started, I just want to acknowledge that the work that I do is integrative and therefore, by definition, really collaborative, and it relies on talented contributions from talented scientists. A lot of the work I'm going to talk about today originated when I was a postdoc in the Nachman lab and continues as we collaborate and our ongoing collaboration. And it also involves the work of some talented undergrads in my own lab at Monmouth University. Of course, I also like to acknowledge our funding sources, and then also just the many collaborators, colleagues, and community members that really form a support network that's needed to do sort of large-scale geographic work like we do. So just to give a high-level overview of the talk today, I'm going to give a little bit of context about the system of wild house mice, and then I'm going to switch over to talking about my work focused on the genetics of environmental adaptation in house mice. And then the last part of the talk, I'm going to spend some time talking about newer work that's definitely in progress. It's a little bit messy on phenotypic variation in these wild mice, but hopefully this will be a part that you guys will enjoy. So you guys, as Emily said, everyone's really familiar with house mice as a model system. And as a genetic model system, house mice have made invaluable contributions to our understanding of the connection between genotype and phenotype, biochemical pathways, and human disease. The list goes on and on. But like other model systems, house mice are real organisms. They exist outside of the lab. They have a unique history. They have predators. They have prey. They have parasites, pathogens, they live in different environments, different habitats. All of these things influence their unique natural history. What is really powerful, I think, about working with house mice is that you can bring a century of research and resources to ask these kinds of questions that are fundamental to evolution, ecology, organismal biology, behavior, and more. And so I think this is a really fertile system just starting to appreciate. So to give a little context on the background of the system, so when people talk about house mice, they're typically talking about sort of this three primary subspecies of house mice: Mus musculus musculus, Mus musculus castaneus, and Mus musculus domesticus. The center for diversity for this group is right around here. And this is essentially where you sort of find Mus musculus castaneus today in India, Pakistan, and Southeast Asia. Mus musculus is so diverged as it moved north into northern China and then into Eastern Europe. And then Mus musculus domesticus diverges as moved through the Middle East and North Africa and then into Western Europe. From Western Europe, house mice spread in association with European colonizers to the Americas, and then also to locations on the coast of Africa, as well as Australia, and a number of oceanic islands. Something I think is really unique and makes them particularly powerful for studying evolutionary genetics is that the divergence among these three subspecies is relatively recent. And what that means is that we can study the genetics of speciation in a group where there is a reproductive isolation to an extent, but the process is ongoing. In addition, the three subspecies have unique demographic histories. So thinking back to Muscus castaneus, it's found in the ancestral range for the group. They have a really large effective population size, which is in contrast to the two other subspecies which have much smaller effective population size. So this is a really good system for asking questions about how this kind of unique history influences patterns of genetic variation across the genome. Oops, there we go. This system has been particularly fertile for studying the genetics of speciation, and so this is a figure from just one of many papers on the genetics of speciation in house mice from 2011, but there are decades of work studying this, everything from the genetics of an introgression across hybrid zones to sort of more organismal approaches looking at things like parasite loads in hybrid zones. These three subspecies hybridize in the wild and in the lab, and hybrid male sterility is polymorphic, and so that provides fertile ground for looking at the genetics of reproductive isolation. And in particular, sort of a large a monumental contribution from this body of work is Jerry Forrett's work after decades of sort of working on the genetics of reproductive isolation, is the positional cloning of PRDM9 in 2009, which is a mammalian speciation gene, which is elusive and really pretty much every other mammalian system. So I hope I've convinced you that the system is a really good one for asking evolutionary questions. It continues to be a system that people are studying the genetics of speciation and asking larger questions about population genetics. And what I'm really excited about is the potential for this system in terms of thinking about the genetics of adaptation. So this is, just a sort of map of occurrence records for Muscus domesticus from GBIF. I just pulled it down this week. I haven't curated it at all, but I think it does give you sort of an idea of the distribution of the species across the world. So domesticus is the house mice that we know and love here in the Americas, but it's also found in this primary range here in Western Europe, sort of its origins, and it's found extensively in Australia and many other parts of the world. So of course, they're living in diverse environments, diverse habitats, diverse climates with diverse pathogens, diverse diseases, different diets. All of this they're doing, and they're doing it quite successfully over a pretty short period of time. And so this provides just a sort of a hotbed of opportunity to think about how these guys adapt to this kind of diversity and diverse environments. So I'm going to switch over to talking about that. I'm talking about the genetics of environmental adaptation. And so that's sort of one aspect of the variation that they encountered as they sort of spread throughout the world is climate, right? And if you're in North America, you're probably familiar with this, but there are really strong latitudinal clines, not only in temperature, but in other aspects of the environment in North and South America. And in particular, there's strong latitudinal clines on the east coast of North America and sort of all aspects of climate. But that's also true in South America, although the slope is a little bit less steep. And the environment directly can affect the fitness of organisms, but through thermoregulation and stress, heat exchange, all of these things. But it also sets up sort of indirect effects through many other aspects of the community assemblages and habitat. So in starting this work, we focus first on the northeast coast and of North America for a couple of reasons. And the first is, just as I said, there's really strong and predictable latitudinal clines in a number of climatic factors that occur over a really short distance on the east coast of the United States. And so there's expectation of strong selection. In addition, there was really nice work by Carol Becker-Lynch in the 80s and 90s identifying clinal variation in body size and also in nest size in these populations and showing that these differences persisted in the lab, suggesting that they had a genetic basis and were not just a result of sort of acclimation to the environment. And both of these traits have pretty clear links to fitness, body size through thermoregulation, and nest size, again, through thermoregulation of neonates. And of course, so this kind of follows the classic pattern of Bergmann's rule, which is just the observation that either animal populations within a species or closely related species, you see this decline and larger body sizes at higher latitudes. And of course, this research has nothing to do with penguins, but they were cute and they're in the Wikipedia article as a demonstration of Bergmann's rule. So I thought they'd throw them in there. Again, north, whether you're going north from the equator or south, the idea is that the more extreme you are, the larger bodied the taxa are. So this suggested to us not only were there strong environmental gradients, but that there was evidence that these traits were under selection. So it was a really good place to start. And so the first sort of transect we did, this is, you're sort of going in blind, other than knowing that body size and nesting behavior might be important. And so we really wanted to be as comprehensive as we could be, given that we were doing a field survey. So body measurements, reproductive status, basically all tissues were taken for either DNA, RNA, or something to do with parasites or status. Urine was collected. Mike Sheehan, who's a professor at Cornell, is interested in major urinary proteins, MUPs, and feces for microbiome work, which was primarily done by Taichi Suzuki, who's now a postdoc at Max Planck's Tübingen. We also created or prepared specimens, skulls, skeletons, and skins for further work. And really importantly, I think, for the sort of future of the system, animals were collected in Edmonton, Canada, Tucson, Arizona, Saratoga Springs, New York, Gainesville, Florida, and Manaus, Brazil, in order to found new colonies of wild-derived mice from the Americas. So I'm going to go through a little bit of what we've learned from the East Coast of North America first, and then I'll talk a little bit more about some of the other transects that are in progress. So we, like Carol Lynch, found that mice from higher latitudes were bigger, heavier, and fatter, and this was consistent than those from lower latitudes, consistent with Bergmann's rules. This persisted in the lab over 2 generations from the wild caught, and then as someone who now has these mice in her lab at 20 plus generations, I can confidently tell you that this is true. Mice from New York and Edmonton are considerably bigger than those from New York and Florida. I mean, from Brazil and Florida. And these differences have persisted in the lab over multiple generations. They also vary with respect to metabolic markers like leptin and adiponectin, triglycerides, glucose. All of these things are good things to help us understand how these mice differ, but also to help us start thinking about making connections between the genetic variation that we find and the phenotypic variation that we see in the field. And what might be especially interesting for this crowd, we confirmed Carol Lynch's work showing that mice from New York build bigger nests than those from Florida. That's also true for mice from Brazil, I believe, although that data is not out yet. And they're also more active. And so this was work spearheaded by Katie Ferris, who's here. And she basically put bike odometers on these wheels and tracked how much distance these guys covered, showing that the mice from New York covered more distance than those from Florida. And I just highlight these pictures here because I'm sure that I don't have to tell you guys, but being able to phenotype these mice, so many mice right when they come in from the field is a really time-sensitive thing that requires a lot of coordination and sort of all hands on deck. And this is Dana Lynn, Katie Ferris, Gabriella Hare, and Taichi Suzuki, Mike Sheehan, who I mentioned before, as well as Mallory Ballinger. All of these folks have been critical in establishing these phenotyping pipelines. So moving on to genetic variation, so what did we observe? So we had moderate coverage exomes and low coverage genomes for 10 mice from each of these sites. We also had RNA-seq data that I generated from mice that had been in the lab for 2 generations, so not the wild caught, but the ones that were in the common laboratory environment. And so we started just by looking at patterns of genetic variation. So this is a genetic PCA, and what you can tell is that mice from the same location, so if we caught these mice in Florida and Gainesville, they're most closely related to other mice from Gainesville, Florida, but patterns across latitude are really idiosyncratic. Okay, so here's Florida hanging out in the middle of PC1, and then flanked by Pennsylvania, and Virginia, with New Hampshire and Vermont and Georgia on the ends. So what this means is that sort of the neutral background patterns of genetic variation in the system are really not following isolation by distance per se. It's much more idiosyncratic. So importantly, what this meant is that we could sort of take two different approaches to understanding how we might be able to identify regions of the genome associated with environmental adaptation. So we use two different approaches. So first we use LFMM, which is latent factor mix model. It's a Bayesian approach to basically simultaneously inferring a neutral structure in the population while identifying outlier candidates. So it's a really useful method, in particular in systems where you have a lot of population structure. But we also just did sort of classic clinal analysis looking for strong correlation with latitude and sort of significant slope across latitudes. And what's nice about that is it's interpretable, right? So you can look and see what the frequencies of that allele are across the latitudes, and you can see that it's highly associated with it, that shift, and helps us sort of focus in on candidates that we think might be contributing the most, or sort of really strong candidates for environmental adaptation specifically. So what we found is that these types of outliers were distributed across the genome. They weren't clustered. It wasn't sort of a single trait or single chromosome or region of the chromosome that was responsible. We also found pretty consistently, both in the genome data and in the exome data, I'm only showing the exome data here, that most of the candidates that we identified were regulatory. They were following in either they were following in parts of the genes that are not coding. So a relatively small portion of them, of these candidate variants, were missense or nonsense variants. And the majority of the genes that had candidate variants did not have a variant that was non-synonymous. And so this really suggested that regulatory changes are important in environmental adaptation. So following from this, we really wanted to use RNA-seq data as a bridge between genetic variation and candidates that we're finding just sort of agnostically by scanning through the genome, to sort of having a proximal phenotype, right, expression. So that kind of helps us get closer to making connections between genotype and phenotype. So we had sort of two different approaches to this. And the first is just looking for patterns of differential expression between males from Florida and New York, the extremes of the clines, and different tissues. But the second was to use allele-specific expression. And what's nice about that is it connects these differences in depth specifically to cis-regulatory changes. And so that kind of more closely ties the differences in expression that you're seeing to changes that are nearby. So we can use heterozygotes to do that. And so what we were able to do is take sort of our larger list of candidates and narrow it down to candidates where we felt like not only were there really strong indications that these were under selection from just looking at our genome scans, but we were also able to tie them to a proximal phenotype in terms of expression. So they had differential expression, allele-specific expression, and the vast majority of these, you can see, have some sort of functional association with body size, fat obesity, immunity, blood chemistry, and one with an association with circadian rhythm. So many of these are phenotypes that vary between the mice, that we know vary between these mice, and some are phenotypes that we are interested in, but haven't looked at yet, like circadian rhythm and immunity. In addition, there are genes that show evidence of selection only in the genome, which is a slightly different sort of survey, but also show evidence of differential expression and allele-specific expression. And I don't want to focus, too much on any of these particular candidates, although other than to say that they're, what I think is really useful about this process is it sort of brings together different lines of evidence to help us narrow down and go from sort of a really large list of potential candidates to ones that we may want to spend more time on in terms of functional connections between genotype and phenotype. But I do like to highlight a few because people usually enjoy that. So this is neuregulin 4. It's highly expressed in adipose tissue. So we can use this functional data that we have from mice to help us understand what's happening here. It's linked to obesity and diet-induced insulin resistance in both humans and mice. In the laboratory studies of obese mice, this neuregulin 4 appears to exert a beneficial effect, reducing the effects of metabolic disorders associated with obesity. And this is also true in humans. And so this seems like a candidate that's worth further exploring further. I just also want to point out, though, so our method is sort of to stack up multiple lines of evidence in order to hone in on candidates that we think are really strong. However, I think it's worth just stating the limitations of this kind of approach is that we're looking at expression at certain time points, right? We're looking at expression in a certain way, in certain tissues, and that's a limited view, right? And so there are other candidates that we identified through the genome scan where we didn't see these sort of differences in expression, but that are really strong candidates based on their status as outliers and that have really strong functional connections to traits of interest that vary in these populations. So a good example of this is MC3R, which is there's lots of data that associates it with things like leptin, insulin, and fat. But in our data set, even though it was a really strong, in fact, I think it might have been the sort of largest, sort of highest outlier in our scans, we just didn't see differences in gene expression. Another approach to this, and this is work that was spearheaded by Katya Mack, was to go back to the transect and actually use the RNA, the tissues that I'd collected in RNA later, and to add to this an additional analysis of allele-specific expression and EQTL mapping of these tissues from wild mice, and identify additional candidates, particularly those that are associated with body size, because that's one of the phenotypes that we were able to measure even in our wild transects. I would just also, I'm not going to talk about the work on the West Coast. This was coming out this week, so I'm just going to plug that this paper will be released later this week in PLAS Genetics. So this work was led by Katie Ferris, and it's a similar approach to what we've done on the East Coast, and has the benefit of being able to compare the results found on the West Coast to that found on the East Coast. So we get to sort of visit the question of parallel evolution. So I'm going to spend a little time, though, talking about this work in progress in South America. So this is work that I'm doing with Felipe de Melo Martins and Jocelyn Gutierrez Guerrero. So we have these samples from continental South America, you can see here, and a couple of sampling locations in Mexico as well. And then Felipe's work on altitudinal transects in Ecuador. There are a couple of lowland populations that we also sort of consider in part of our environmental adaptation survey across latitude. So we have these different samples. I don't want to go too deeply into the details of all the different sort of ways we're looking at this data. I'm sure you guys are familiar with sort of analyzing things in lots of different ways, other than to say that our approach was pretty similar to what we did on the East Coast, which is that we focus on exome capture sequencing. We had pretty good coverage of about 35x, and we were using LFMN to identify outliers. In this case, and I'm going to show you why, we're not using a kinal approach in addition to that, and you'll see in a second. So South America has a latitudinal decline in climatic variables. I'm showing here annual mean temperature, but you can actually do sort of a PCA analysis of the climate variables that people pull down from bioclim. There's a really strong association with the first principal component of those and latitude. So it's still a really strong driver of variation in climate, but you can see the slope is a lot gentler in comparison to North America. So the shift in temperature in South America occurs over a much larger number of latitudinal degrees. And the difference, you can see, it sort of occupies different temperature space, right? So it's definitely colder in Tierra del Fuego than it is in equatorial Brazil. But we're sort of already starting a little higher up on the temperature scale, and the shift is a little bit less dramatic. In addition, there's really strong indicators of isolation by distance in South America. So just in this first graph, I have them sort of color-coded, but just to make it really easy for you guys, you're essentially going from the southern tip of Argentina to equatorial Brazil. This is, the first principal component of genetic. Then the genetic PCA essentially recapitulates latitude. And so this is telling us that there's a strong association between geographic and genetic distance. And just to put this in context, this is all of the populations that we have in South America and Mexico added into North America. So you can see there's a pretty large break here on the first principal component that's splitting apart North America from Central and South America. And then on the second principal component, we're sort of spreading out Central and South America by latitude. So what this means is there's a lot of isolation by distance. We can't really use kinal analysis because the whole genome's sort of acting in that way. So we're focusing on using LFMM to identify candidates. And much like we found on the east coast of North America, the candidates are distributed across the genome. There's no clustering. We're focusing on a relatively short list of outliers, about 175 genes. And these candidates relate to traits, again, like body size, adipose, immunity, behavior, metabolic disease, and interestingly, tail and ear size, which is something that is found to vary in these mice. Again, just like with the East Coast, most candidates are not amino acid changing, but it is true that outlier SNPs are enriched for non-synonymous and UTR SNPs, which indicates that they're sort of more functional hits, and that wasn't the case in the East Coast. And roughly, depending on how you do the analysis, 20% of candidate genes have a candidate non-synonymous SNP, which means that on the other hand, about 80% don't. So regulatory evolution, again, seems to be critical. Just again, to give you a couple of candidates. So this is androgenic receptor beta 3, ADRB3 is the abbreviation for it. There's a mutation that's amino acid changing that's a candidate variant identified in the scan. Phenotypes in these knockout mice relate to polyphagia and susceptibility to weight gain, increased body fat, body weight, and other phenotypes associated with body size. This was also identified as a candidate in the North American transect. The second one here is calveolin-1, or CAV1, as it's abbreviated. And then this is a functions in the uptake and regulation of fatty acids. Knockout mice show changes in adiposity, resistance to diet-induced obesity. It falls in QTLs related to body size. It was identified as a candidate in the East Coast mice, and interestingly, also identified as a potential candidate affecting body size in the Gulf Island mice. And so these mice are found in an oceanic island. They are giant, they're exceptionally big, and have had a switch on diet to eat seabirds and seabird eggs. So it's really interesting that we have a candidate that shows up in so many different studies. In addition, there is evidence of shared response to selection. So this is just a comparison with the East Coast of North America. There is a lot of overlap in terms of the candidates that are identified, regardless of which approach you take to sort of calling something a candidate, and it's more than expected by chance. So this suggested that, again, that there's some common response to selection. On the other hand, the top candidates in each transect are different, which is maybe not surprising. And this suggests differing selection pressures or differing responses to selection on the among the transects. One of the other things that we're exploring is essentially analyzing all of the data together and identifying unique candidates that we find only when we sort of consider all of the data, both North and South America. And in fact, there are candidates that arise, things, again, related to things like body size, metabolism, immunity, and other really interesting phenotypes. So just to summarize this section of the talk, so we find genetic and geographic distance to be highly correlated in South America, but not so in Eastern North America. We find that regulatory sites are dominating candidate SNPs, but there's enrichment for non-synonymous and UTR SNPs among extreme outliers in the South American transect. A lot of good candidates for future research using multiple methods and transects, and there's overlap among the transects, suggesting some common response to selection, but also some unique responses. We're going to switch over to talking about phenotypic variation, and part of that is because these big surveys are really, really valuable in terms of understanding what's happening across the genome, but they also produce hypotheses, right? And so there's only so many phenotypes that we can measure in the field, and there are only so many phenotypes that we sort of guess in advance that might be interesting to consider and that are tractable to consider when we think about how we're sort of going out and collecting mice. But the fact that we were able to bring some mice back into the lab and start new strains provides an opportunity to follow up on some of these phenotypes in a more comprehensive way, and also to explore some phenotypes that we might not have anticipated as being interesting, but that might come out of these scans. So, like I said earlier, this part of the talk is pretty rough and speculative, lots of new data, but I did want to include it, especially because it represents work that's done by undergraduates in my lab and under pretty, stressful conditions in the past year. And so I really wanted to honor the effort that they put in despite all of the challenges that we've had and show you sort of some of what they've been able to find. So I'm going to focus on reproductive traits and then also sort of revisit body size. So, of course, reproductive traits are fitness, right? They are the definition of fitness. They're really hard to measure in wild populations, especially in house mice. It's really hard to get the numbers that you need to understand what kind of variation there is. But we do know that so from our data, we know there's a strong association between body size and latitude. And from a lot of other data that's been collected in the past, we know that small mammals, larger females tend to have larger litters. We also know from other studies in the past that there's some evidence of larger litter size at higher latitudes in small mammals. And of course, this isn't something that we could measure in the wild when we were doing the transects, but it is something that we can sort of go back to and think about now that we have mice in the lab. So another sort of approach to this, or sort of theory behind this, is that, we have mice from Manaus, which is a pretty continuous environment, right? It's kind of aseasonal in terms of temperature, although there are rainy seasons. And that's in comparison to somewhere like New York, which is strongly seasonal and where there may be relatively limited opportunity for reproduction. And so there's sort of this potential life history trade-off between having multiple litters throughout the year and conditions that support that, and having maybe 1 or 2 chances at most a year in order to have a litter. And so in addition to the connection between litter size and body size, there's also this idea that the seasonality of an environment may influence sort of the size of the litter and the number of litters per year. So this is Tiffany Longo. She was an undergraduate who started with me as a freshman, and she was really interested in life history questions, and she proposed that we go back through the breeding records just to look to see if litter size was different among these locations. And the results of that were promising enough that when we started our new colony here in 2018, which took quite a while to set up, we started to take a really much more structured approach to looking at litter size. And we also weighed pups at 7, 14, and 24 days, and of course the litter weight. And then we started also to track the weight of our dams at weaning so that we had a standardized weight for the mothers. So just going to show you some of the analysis that Tiffany has produced. So she's found that their location of origin contributes significantly to variation in litter size. And these are all from early generations after the wild-caught animals were brought into the lab. So really the first and second generation of breeding. And you can see here that, these data, of course, are ordinal. You can only have whole pups. So you can see that creates some fun bee swarm plots. But the post hoc test finds significant differences between Edmonton, for instance, and the other locations, as well as New York and Florida. So this was promising enough, like I said, that we sort of decided to keep looking. And in later generations of inbreeding, now we're focusing on specific lines because we're past 10 generations of inbreeding, which is where significant portion of the genome is now homozygous. And so we have 2 strains here that we're looking at from Edmonton, 2 from New York, 1 from Arizona, 1 from Florida, and 1 from Brazil. And you can see there are differences that location contributes to variation in litter size. And in particular, these comparisons between 82 by 81, which is a New York strain, 1 from Florida, and then also from Arizona, are driving most of the differences in these comparisons. So now that we have them in our own lab, we can do things in like I said, a sort of a more structured way. And so what we're basically doing is counting pups at 7 days, weighing those pups at 7 days, and then following up and weighing them again at 14 and 24 days. And here you can see there's 1 strain from Edmonton, 2 from New York, 1 from Tucson, 1 from Florida, and 1 from Brazil. And litter size tends to be smaller in Florida and Brazil than it does in the other populations. Pup weight tends to be smaller in Brazil. And then you can see this 1 New York line as well. And then if we sort of take those 2 factors together, you can see that overall the sort of reproductive investment in the first litter from these moms in Florida and Brazil is lower than from the other populations. And I just want to, it's important to note here that we're using the weight of the mother at weaning as a covariate in these analyses, and location is still strongly significant. So one of the other things we're following up with is actually looking at their maternal care. And this is Carolyn Reverindo, and she's spending a lot of her time watching videos and using Boris to score how much time that these new mothers spend attending to their pups, and just get a fuller picture of the reproductive investment and the potential differences between the strains. And I just want to put a plug in here for more behavioral analysis. So the Jackson Labs is rederiving these strains, and so they are doing sort of a standardized phenotyping pipeline. But there are many potential questions that could be asked with respect to behavior in these systems, many of which are sort of outside of the standard pipeline, things that have to do with exploratory behavior, risk avoidance, stress tolerance, social behaviors, flexibility, diet choice. And there's a nice tradition of this kind of work in wild house mice, despite the challenges of doing it. So I think it's really something that is worth spending time on. I'm just going to show you a little bit of data from Jesse. So Jesse used these activity chambers to do some of those very sort of classic studies of light-dark tests and open field tests, just to get a feel for the differences between the population. She, like the other students, was really impacted by the time constraints of COVID. So we have relatively limited data, but surprisingly significant, showing really differences between behavior from mice from Brazil, Edmonton, and New York, both in things, here we're just showing sort of total distance covered, but also in terms of things like exploratory behavior and time spent in refuge use. For this last part of the talk, I'm sort of going to go back to this idea of body size and the genetics of body size and sort of the genetics of Bergmann's rule, right? Understanding why it is that these mice at higher latitudes are larger. And one of the things that I think is really interesting to think about is, when we do these studies in the field, we're sort of getting one moment in time. And then when, by bringing them back in the lab, we can change the context. So we can ask about how changes in the diet affect these kinds of traits. We can ask about how it affects growth curves, body size. We can look at the gut microbiome. We can look at effects of sex, which is something that was something we could only do in a very limited way before. So it opens up a lot of doors in how we a richer idea of the phenotype. And in particular, I'm interested in patterns of gene expression and how we can use them to tie these genetic variation to phenotypic variation. And so what we've essentially done is a controlled experiment where we have these mice growing up on regular breeder chow, but also on high-fat chow, and just measuring them regularly over the course of 12 weeks. And so I just want to show you this too. This is sort of hanging over from the past, looking at the pup experiment. Part of the reason why we started to think there may be differences in their growth rates, for instance, is that when we look at 24 days, at 7 days, this MJS 19x13 strain is relatively small, but they've really rebounded by 24 days. And at 24 days, you see this sort of clear delineation between the Edmonton and New York mice and basically everyone else. And this is consistent with what we find in the field when we look at these traits. And so this suggested to us that there are differences in growth among these mice, and that's something that might be worth looking at. And so again, this is a little bit of a messy slide. It's new data and new analysis produced by Emily Tobacco and Lorena Mancino. But you can see we have sort of females on one side and males on the other. We have New York and Canadian mice here, Florida and Brazil here. There are differences in how they respond to the high-fat diet. And there's also sort of differences in sort of the shape of their curves. And in particular, we're interested in the interaction. So the differences in the way that these strains react suggest plasticity, right, in response to their diet. And we find that with using ANOVAs with wean weight as a covariate, that there's significant effects both of diet, strain, and the interaction between diet and strain, which again tells us that there's this plasticity in response, which I think is really fertile ground for dissecting out the connection between gene expression and genetic variation and the trait itself. So we have another sort of subset of this. We're running an experiment where we're doing all of the things I just said and then looking at differential expression in the tissues that are related to metabolism, but also doing a cross between the two that allows us to ask questions about allele-specific expression. So we're able to look at differential expression and allele-specific expression as baseline on a normal diet, but also the differences that are diet-induced. And the hope of this is that we're better able to connect genotype and phenotype using these patterns of gene expression. Okay, so just to sort of summarize all parts of the talk today, in terms of wild house mice and just population genetics and evolutionary genetics more broadly, this work is starting to help us understand a little bit about population structure in the Americas. And there's a student in Michael Nachman's lab, Kennedy Agwangba, who's going to be doing sort of a massive population genetics work with all of these populations. And then from the environmental adaptation work that we've done, we find loci across the genome, signatures of selection, both in North and South America, strong evidence that regulatory evolution is important in these systems, also shared and unique responses across the latitudinal clines, as well as really good candidates for continuing functional analysis. Finally, we have we've been able to relate phenotypes that are important to fitness and that vary predictably with climate and persist in the lab. And we're identifying new additional phenotypes that vary among these strains that I think will be really fruitful in the future. What I think is the most satisfying part of this kind of work and really gratifying is that not only are we sort of asking and answering the questions we're interested in, but we're building a massive data set and resource for the community. So we sort of we've used all the resources from the genetic model system, and now we're building up resources for wild house mice. So we have genomes and exomes and transcriptomes, we have natural history data. And I think that all of these things are going to provide a foundation for really interesting work in the future, both for me and my collaborators, but hopefully for the broader community. In addition to this, the Jackson Labs, like I said, is rederiving some of these wild strains, and then they'll be available for the community to work with in the future, which is, I think, really exciting. So I'm just going to wrap up and take your questions. I also just want to put a plug in here that there's the whole sort of side of my lab that works on marine genetics, and lots of students are excited about that, and that's something that we, as a university on the ocean, are really excited about. Okay, happy to take any questions from the crowd. All right, so I can see some of the questions. So from Sarah West, the question is, did house mice replace native species in the Americas, or do they occupy their own niche? And I'm going to try to stop sharing my screen. There we go. Okay, so you guys can see me. And the answer is, that's a good question. Okay, so in much of the range where you find house mice, you also find Paramiscus species, deer mice, and things like that. And I do think they compete for space, and also with rats. So you may or may not know that rats actually predate on house mice. And so there is a tension in terms of use of, like, for instance, like barn spaces between the native Paramiscus, then the exotic house mice, and also the exotic rat species. And in fact, in some cities you go to, and places that you might think would be house mice locations, they in fact have Paramiscus. Hopefully that answers your question. So I do think they compete, and I think in some cases they may replace fringe habitat for things like deer mice, right? So deer mice might have invaded a barn in the winter when it's cold. Can't do that if house mice are occupying it. Although I don't think that house mice are always the winners in that. Do you have a hypothesis for why mice in more extreme latitudes are more active? Maybe to prepare for resource gathering in a wider area? Yeah, I think the answer to that is probably pretty complex, and probably involves both metabolism and behavior. And so I'm not even going to try to answer exactly why. I'm not going to try to give you a definitive answer on that. But I do think it may have to do with basal metabolic rate and differences between the sort of thermoregulatory differences between the mice. May be condition dependent as well. So Mallory Ballinger, who I mentioned in the talk, is really interested in the sort of plasticity of some of these traits in response to temperature, and has done some experimental work on that. So look for that. But I do think that there are potentially also behavioral explanations for activity, and especially for things like exploratory behaviors, and there are differences in predators, for instance, potentially, or for availability of resources. Lots of unanswered questions, though. Okay, next question. And this is from Emily, I think. It says, I'm curious about the genetic and geographic distance differences among North and South America. Do you have guesses about population mobility or movements that may relate to this extreme difference? Yes, I do. So first of all, when we think about the Northeast, the sort of Northeast in North America, 95 runs the route, like runs right up from Florida to the Northeast. And so the connectedness between Florida and New Hampshire, just sort of a mobility, I expect, is quite high. And remember, these mice, if they're traveling amongst states, they're probably traveling on trucks with feed or hay or something like that. And so I think there's probably quite a lot of mobility, and there's no political boundaries, like in terms of countries, right? So there's state boundaries, but not like there are no political borders along there. So that increases human traffic across those runs. The other thing is the geographic distance is actually smaller on there compared to South America. So South America, on the other hand, we have a lot of political borders. We have a lot of topography as well. So we were very careful in these populations to not sort of confound altitude, but you can't not think about South America and think about the altitudinal differences that actually might prevent movement between populations. So I think there's probably much more restricted movement between populations in South America than in populations in the East Coast of North America. All right. I think that's all of the questions. If anybody else wants to add one in? Okay, so I guess we're having some AV issues, so I have the privilege of sort of closing this, and I'll just say again, thank you so much for joining and listening to the talk today. I was very happy to join your group. And if anyone has any questions or wants to follow up and discuss my work or your own work in relation to it, please shoot me an email. I'm happy to chat with you. Thank you.